Chapter 2, Section 10, Surface Motility. Now, surface motility, when we talk about this, we're not talking about a microbe swimming through a mud puddle. We're not talking about a microbe swimming through your bloodstream. What we're actually looking at is the ability of some of these bacteria to move when it's on a surface, like on an agar plate or whatnot. This motility we're going to see here, known as twitching or gliding, it's much slower than swimming. It's going to be driven by other appendages that are not flagella. We can see the, as here these microbes, as things are moving, they're stretching out, they're expanding out from the center here. I forget what these two are, A and B. We're not going to worry about that right now. Let me see. The cyanobacteria here, they can use what are known as gliding. This allows them to move across the surface of wet, or across, the top of wet surfaces. It's a very slow process. First one we're going to talk about is what is known as twitching. Okay, twitching requires those type 4 pili. Okay, we've talked about these before. These are going to be, projection appendages outside the cell, hollow appendages that allow for adherence, allow for the transfer of. And here it's going to allow for some movement, because remember the pili can move like this a little bit. Not the spinning like we talked with flagella, but there's a little bit of a movement, a little bit of a lateral waggle. They extend from one cell, attached to a surface, and then they will be retracted back into the cell. So that waggle and then pull back in allows them to pull the cell across the surface. This is ATP hydrolysis driven, okay? So it's ATP driven. This is not going to be a proton motive force thing. We see this with Pseudomonas. Pseudomonas is one of these interesting bacteria you go through and you spread it on a plate in a certain way, so you end up with certain colonies, and you can literally watch, go back every few hours, every for a couple of days you do this, and you can see as that colony expands, and it will expand well beyond what you think it should, and it's almost like rings of growth, as the older bacteria kind of die off and the younger bacteria spread a little more, and then the next and the next and the next, forming these rings, this progression rings. You also end up with what is referred to as gliding motility. This is a smooth continuous that does not use external structures. Very few of these. These are going to be bacteria that do this kind of movement across. They have proteins and whatnot in them that allow for them to contour the body shape a bit. So that twitching, extend out that pilus-like thing, it attaches, it retracts, and it pulls the bacterial cell. Whereas this gliding, it's like I said, it's more of a contortion of the cell. So it kind of spins, and as it's spinning, it's moving.